Hello students, here we are going to discuss another important, very important concept, that is enzymes. What are enzymes? Enzymes are nothing but the biocatalyst or chemicals. They are nothing but proteins. Simply I can write enzymes as proteins. Okay, so these are also a type of proteins. Remember, not all the proteins are enzymes. Got it? Okay, but all the enzymes are 100% proteins. Even hormone is a type of protein. Okay, fine. So the definition of enzyme, let me just write the definition here. These are a very good biocatalyst. What is the meaning of biocatalyst? Catalyst is nothing but a type of substance which actually helps a particular chemical reaction, so that the chemical reaction takes very less time to get complete. Bio means life, so I'm talking about the chemicals which is very essential for life, which is actually catalyzed by this type of protein called as enzymes. For example, one simple example, A plus B is a type of two chemicals. We have two chemicals, so which undergoes chemical reaction to produce a product. What is the product here? AB is the product. Okay, let us assume that this entire reaction takes about 15 minutes. It's a long time actually for a very tiny chemicals to produce the products. 15 minutes is very a long time. If I just add one enzyme here to this reaction, so they actually catalyze this reaction, and then ultimately the product, that is AB, is produced. And for this we need hardly 0.1 second. See, without enzyme a chemical reaction takes longer time. With enzymes chemical reaction takes very less time. This is actually the advantage of chemicals. In our body we have millions of enzymes. Got it? They are classified under 6 major groups. I'll talk about it later, that is a classification of enzymes later. Okay, so you understood what is enzyme. Now let me just write this. Enzymes always speed up the chemical reaction. They actually speed up the chemical reaction, but this point to be noted, they cannot initiate a chemical reaction. They cannot actually start a chemical reaction. Got it? When the chemical reaction is running, an enzyme can come and speed up the reaction. Got it? But it cannot initiate a chemical reaction on its own. This is very important word. I just have to write this. Very important. Please note this. Ribozymes. What are ribozymes? Very important for your competitive part. Ribozymes, these are actually not enzyme, but they work like enzymes. Got it? They are not enzymes actually. The definition is very simple. These are nucleic acids. So what are ribozymes? Ribozymes are nucleic acids. Got it? They behave like enzymes. They are not enzymes actually. They behave like enzymes. Nucleic acids. What are nucleic acids? DNA and RNA. We had already discussed about nucleic acid in our previous video session. Yes? Okay, ribozymes are nucleic acid. They behave as if they are enzymes, but they are not enzymes. They also involve in a chemical reaction and speed up the chemical reaction. Got it? Okay, fine. So next one, let me just write the structure of enzyme. This is very important. Structure of an enzyme. Okay, hope you are getting my point now. Normally we know that the enzymes are made up of polypeptide chain. The enzymes always tertiary or quaternary in structure. Okay, so they are like highly coiled polypeptide chain, maybe made up of many polypeptide chain. So the 3D structure of the enzyme looks like this. See, it looks like a C, right? Let me just draw in another form. It looks like this. Got it? And the specialty of this enzyme is it has got a pocket-like structure. You could see empty space here, a cavity, a pocket-like structure. This pocket-like structure is called as active site. Got it? So this is active site. So what is the function of active site in an enzyme? Very simple. Normally enzyme actually comes to a chemical reaction, and then actually it speed up the chemical reaction. For that it has to bind to a chemical. The enzyme should bind to the chemical, got it, to produce the product. So the product is formed from that chemical, but it is initiated by the product formation is initiated by the enzyme when a substrate binds to the active site. So here let me just draw. This is the enzyme. Now this is the active site, and I'm using the red color pen, and this small box, okay, so this is now a chemical, okay, the chemical which is undergoing chemical reaction, and here we just name that chemical as substrate. Okay, in other words, it is the food for the enzyme. That's it. Okay, a chemical which binds to the active site of the enzyme, and ultimately you get a structure which looks like this, see, when it binds to the active site, see, the red color block is already fitted. Got it? It fits to this active site, and now see, this is enzyme and this is substrate. When enzyme binds to the substrate, it is called as ES, that is enzyme substrate complex. Okay, so this is very simple now. And now once enzyme actually binds to the substrate, it takes some time, a fraction of second actually, not more, or time, not much time actually. Okay, so ultimately this ES complex undergoes a state called as transitional state or transition state. What is this transition state? Transition state is nothing but when a substrate binds to the enzyme, it undergoes structural modification. Here enzyme is undergoing structural modification or substrate. Actually substrate is undergoing structural modification. That is, the bonds present, the chemical bonds present in the substrate is broken down. Very simple. For example, if my hand, this is actually enzyme and this is the substrate. Substrate now fits to the enzyme active site. And now see, I have got 5 fingers here, the substrate, the chemical bonds. See, now what's happening? It's broken. And ultimately, see, got it? This is called as transition phase. What is transition phase? When enzyme binds to the substrate, it actually breaks the chemical bond. It takes some time, okay, because of the, it binds to the substrate very tightly. So it brings structural modification in the substrate, and ultimately substrate, now the chemical now becomes products. Got it? So this is what actually transition state, and ultimately after a product is formed, enzyme now separates from the product. Okay, so now you could see the dot here. These dots are nothing but substrate. After transition state, it forms product. So I just write P here. So this is E. Got it? Enzyme is now free, and again it binds to another substrate, and ultimately it undergoes ES complex, then transition state, and ultimately products are formed, and the cycle is repeated. So this product is now formed. Done. So this is the simple mechanism of action of enzymes. How exactly enzyme work. So you understood how enzyme look and what is active site, how it binds to the substrate, and how a product is formed. Simple. I can just give you very simple example, that is in glycolysis, which is the first substrate which undergoes glycolysis to produce pyruvic acid, the glucose, correct? Okay, so glucose is now nothing but, in this case, substrate. So the first substrate in glycolysis is glucose. Now the glucose binds to one enzyme. Can you please name the enzyme which converts glucose into glucose 6-phosphate? Yes, that is hexose, correct? Hexose enzyme actually binds to the glucose, and actually hexokinase, not hexose, hexokinase, sorry. Okay, this hexokinase now changes the glucose, that is the glucose is now undergoing transition state, and ultimately a product is formed. What is the product? Glucose 6-phosphate. Done. Got it? Even glucose can undergo certain transition phase without the help of the enzyme to produce glucose 6-phosphate, but it takes much and or more time. Okay, so it's time consuming process. It could be 20 minutes. Simply, if I add hexose kinase, that is hexokinase, to this glucose, ultimately it produces glucose 6-phosphate, and for that it takes hardly 0.1 second. That's it. This is actually the advantage of having enzyme. It speed up the chemical reaction. Done. Okay, I give you very good example, that is it's even mentioned in your textbook, that is carbon dioxide. When it binds to water, what is the product is formed? Here I'm not adding any enzyme. Okay, without enzyme a new product is formed, that is called as H2CO3. What is H2CO3? Carbonic acid, correct? Yes, carbonic acid, right? Okay, and here let us assume that within 1 hour, without enzyme, it's going to produce 200 molecules of H2CO3 without enzyme. Got it? Carbon dioxide and water ultimately produces 200 molecules of carbonic acid per hour for 1 hour. Okay. Now for the same reaction, if I add the enzyme, what is the enzyme actually for the production of carbonic acid? That is, you know that carbonic anhydrase. This enzyme got the highest turnover number. You have already studied, correct? Okay, in that is breathing and exchange of gases chapter. Carbonic anhydrase produces the same product, that is H2CO3, that is carbonic acid. And let me just tell you, per second, just for 1 second, how many molecules of carbonic acid is produced when carbonic anhydrase is actually initiating or taking this chemical reaction. That is around 6 lakhs. 6 lakhs of carbonic acid molecules are produced when carbonic anhydrase takes care of this reaction. That is, in the presence of this carbonic anhydrase, 6 lakhs of carbonic acid is produced. That is this per second. Got it? So you know the main significance of enzyme. Very simple. Done. Now let us just assume one more thing, that is you take this chemical reaction. Okay, for any chemical reaction, okay, when an enzyme is present taking care of the chemical reaction, okay, so we need to remember this formula, that is rate of the reaction can be calculated by a simple formula, that is delta P divided by delta T. So what is this rate of reaction definition now? Very simple. It is the number of, P means products, number of products formed per unit time. So what is this unit time? That is given time. For this reaction, 1 second is the unit time. Got it? How many products are produced? 6 lakhs. So can I just write this 6 lakhs divided by per unit time, that is nothing but 1 second. So this is actually simple to calculate the rate of the reaction. Got it? Okay. So one more example, if you want, you can also take this. Glycolysis normally contains 10 enzymatic reaction steps. Yes, right? And you all know the different types of enzymes: HIFI, DK, MEC. So how many are there total? 5, 5, 10. 10 enzymes actually take care of one particular chemical reaction, that is glycolysis. This is the basic introduction of enzyme. Remember, there are so many factors which affects enzyme activity. Whenever you get fever, simple fever. So normally the body temperature is 37 degrees Celsius. When you get fever, automatically your body temperature, that is your hypothalamus in your brain, increases your body temperature to more than 37 degrees Celsius. It could be 38 or 39, sorry, not less, 40. Okay, see, the body temperature is increasing. That is, the internal environmental conditions are actually, particularly the temperature is high. So enzyme automatically stop working. This is one of the reason why whenever you get fever, you get tiredness, because enzymes are not working properly. Got it? It's a simple reason actually. Okay, so when your body temperature reduces below 37 degrees Celsius, again enzyme activity stops. So enzyme require a particular temperature, that is, what is the optimum temperature required for enzyme to work inside your body? That is between 36 to 37. Okay, so between these temperature the enzyme work. Got it? Okay, so you need to maintain this temperature. Obviously your brain, the hypothalamus, will be taking care of this, don't worry. Okay, but when you get fever, automatically the enzyme activity reduces and automatically you get fatigue, that is tiredness. Simple. Okay, now let me just go to the next concept of the same major concept, that is enzyme. That is the mechanism. I have already discussed, but let me just go with the concept. Mechanism of enzyme action. How exactly enzyme work? Simple. For example, we'll take a black color. Step number 1, this is now enzyme. Okay, enzyme got active site. What is active site? It is nothing but because of the structural changes when a different polypeptide chain, many polypeptide chains are actually linked in a protein, that is enzyme. Obviously it makes a pocket like structure, and this is called as active site. To the active site a chemical binds. The chemical is nothing but here, that is substrate, that is substrate, and this is enzyme. And what are you going to get now? You are going to get a different structure, that is binding of enzyme to the should I use red color? Okay, to the substrate. See, this is it. And what you call for this, you call it as enzyme substrate complex. Simple, easy. Are you getting my point? So this is actually the first type of any type of enzyme action done. Once it is done, the next is this particular enzyme, that is substrate complex, undergoes a kind of like a state that is called as conformational change. It is also called as transition state. Okay, it varies. If the chemical is very complex, then obviously transition state requires a lot of time. If the chemical structure of that substrate is very simple, then it takes very less time actually. That's it. That is actually transition state. Got it? Okay, so please remember this. Transition state takes some time. Got it? What is this transition state? It is nothing but time taken by the substrate, not the enzyme actually, substrate, to undergo structural changes. That is nothing but bond breaking, structural changes, because the bonds are present within the substrate has to be broken. Got it? Otherwise it cannot produce products. So this is transition state. Okay, so once transition state is achieved, the third thing is, let me just write, this particular enzyme now breaks the bonds in the substrate, and now the substrate automatically becomes products. These dots are now products. That's it. Simple. Got it? Okay, so the products are finally formed from the substrate. So this is how the enzyme works. Got it? Enzyme first binds to the substrate, then it brings conformational changes in the structure of the substrate, that is called as transition state. It takes it depends actually what type of chemical is the substrate in this case, and ultimately the products are produced. And now this enzyme is free. What is the function of this enzyme now? Again actually it undergoes the first step of this enzyme action reaction, and it binds to a new substrate, and again the same will be repeated. This is how it works. Got it? Okay, so hope you got this now. Simple. Please understand what is transition state. It is the time taken by the substrate to undergo conformational or structural change, ultimately to produce the products. This is transition state. Got it? Okay. Transition state is seen only when enzyme is bound to the substrate. When the substrate in its free state, free form, when it is not bound to enzyme, we cannot call it as transition state. Transition state can be called when a substrate binds to the enzyme and undergo certain changes here itself. Got it? So this is actually called as transition state. It's very simple. You take lock and key. So here the lock is now enzyme, and it has got a small hole actually, that is now active site, and substrate is nothing but the chemical, is nothing but the key. When you put inside the lock, and then actually you start turning, correct? Okay, when you just turn the key, automatically the lock gets open. So when you turn, the time taken to the time taken is called as transition state. This is it. Got it? So that's actually the thing. Mechanism of enzyme action is done now. Okay, now I'm going to talk about another important, very important concept, that is transitional state, or it is also called as activation energy concept. This is very important. You need to remember a graph, that's it, because I'm teaching this for your competitive part, so no need to mug up. Just very simple. Activation energy concept. What is this activation energy? Okay, so let us just learn that. For that I'm going to draw a graph here. Please draw this graph. So this is our x-axis and this is our y-axis. Okay, Now let us just assume that this line is now undergoing chemical reaction. Okay, this line is nothing but now the chemical, what you call for the chemical, you just call it a substrate. Yes, right? Okay, very simple now. Now I'm using the blue color pen. If this substrate has to undergo transition state to produce the product, okay, without enzyme, how the graph looks now when the enzyme is absent in this chemical reaction. So let us just assume that this is the starting point of the substrate which is now undergoing transition state without enzyme. See, and ultimately here the products are produced. Simple? Yes. Okay, see from here, this is actually very simple. This is the potential energy of the substrate. Okay, this is the potential energy. So let me just write the y-axis represent potential energy. Okay, and this one, the x-axis, represent progress of reaction. Yes. Okay, so potential energy, progress of reaction. So the reaction is going now. This is the potential energy, the state where exactly substrate got its potential energy. Now this substrate undergoing chemical reaction, that is this point, this is the peak actually, where ultimately the bonds are broken in the substrate without enzyme, and this is the highest peak, and that is called as transition state. You know what is transition state now? It is nothing but time taken by the substrate to break its bonds to produce products. Okay, so this is the highest peak. Let us just put a line here, put one arrow mark. See this arrow mark now, it's very long now, correct? So let me just label it the chemical reaction. Okay, or I can simply write this as activation energy. Please understand what is activation energy. Activation energy is the energy of the substrate, that is nothing but potential energy of the substrate to break its bonds. Okay, and in this case enzyme is not there, so activation energy without enzyme. Hope you got this point. Okay, so substrate got its potential energy now. Okay, it is stored actually, and now substrate undergoes chemical reaction, and ultimately the bonds are broken by itself without enzyme. Enzyme is absent completely, and ultimately during that process it undergoes like certain transition state. What is transition state? It is the time taken by the substrate to break its bonds. Done. Okay, sorry for the disturbance again. Okay, so ultimately this is actually the graph, activation energy without enzyme. Now for the same substrate, what if I add a chemical, enzyme, okay, for the same substrate. Okay, so this is the difference now. You see now the same products are produced when I added the enzyme. So activation energy, this second graph, with enzyme, when I added the enzyme, the activation energy required to break the bonds is very less, right, compared to the first graph. Yes. Okay, so you could see the difference now. See this arrow mark and see this arrow mark. This one without enzyme, this one with enzyme, very less. That's it. So when you add the enzyme, obviously it reduces the activation energy of the substrate to break its bonds. Got it? So this is the another advantage of enzyme. So that is actually activation energy concept. Please remember two things here. Let me just write the first one. Activation energy, we add the enzyme. So very simple. Sorry. That's it. Activation energy required for this particular chemical reaction is very less when I add enzyme. But now activation energy requirement is more when you don't add the enzyme to the chemical reaction, more when enzyme is absent. That's it. This is actually activation energy concept. Very simple. It is nothing but the difference in the activation energy required when an enzyme is absent or when an enzyme is taking care of the reaction. That's it. So what is activation energy? Activation energy is the difference in the potential energy of the substrate when an enzyme is present or absent. That's it. Got it? Difference in the potential energy of the substrate when an enzyme is present or absent in the chemical reaction. In the chemical reaction. So this is actually called as activation energy. Got it? So what is this activation energy? Activation energy is nothing but the potential energy of the substrate. Okay. What is the potential energy required to break bonds in the substrate to produce the products? Got it? When you add the enzyme, obviously activation energy requirement is very less. When enzyme is absent, activation energy required to break the bonds in the substrate is high, more. Simple. Got it? So this is what actually the thing you need to understand. Okay. So this is all about activation energy. Now we will discuss another very important concept, that is factors affecting enzyme activity. Hope this concept is very clear. Okay. Fine. Let me write that factors, very important concept, factors affecting enzyme activity. Okay, don't worry, it's very simple, it's not that complicated. Only thing is you need to understand the concept. Okay, what are the factors which affects the enzyme activity? Okay, so there are actually four factors. Let me just write the first factor here. Anyhow, I'm going to discuss in detail. The first one is temperature. More or less temperature, obviously it's going to affect the enzyme activity. Okay, so the first one. Second one is the hydrogen ion concentration, the pH. Okay, each enzyme requires optimum pH. If it goes less or high, obviously enzyme activity shuts down. Okay, next one is there are so many chemicals which actually competes with the enzyme's active site. Instead of actually, when enzyme binds to the substrate, obviously we know that chemical reaction takes place. Okay, if the substrate is replaced by another chemical which is not a real chemical, so then the chemical reaction takes a different route. That's it. Got it? You are not going to get the desired product, you are going to get a different product. Okay, so that is nothing but inhibitors. The next one is substrate concentration of substrate. I'll tell you what is this concentration of substrate. These are the four important factors which is going to affect our enzyme, any type of enzyme. Okay, so let me just go with the first one, that is temperature and pH, because both are same. So I'll just write the first factor which affects the enzyme activity is temperature and pH. So this is what very important factors. Okay, the easiest way to understand this, see, if this is the enzyme and this enzyme got a particular pH as well as temperature, so it works only when this specific temperature is provided. In our body it is 36 to 37 degrees Celsius. Okay, and even the pH also, the pH should be between 6 to 7. Okay, so not exactly 6, it could be like 6.5 to 7. Okay, so this is the optimum pH for the enzyme in our body. Temperature should be 36 to 37 degrees Celsius. This is the optimum pH. Okay, if there is, okay, so let me just write that. That means it is understood that enzymes work at optimum temperature. Please write this, temperature as well as pH. Optimum temperature and pH is required for the enzyme to work. Okay, so this is one thing. And second important thing is the activity of the enzyme decreases. Let me write first, activity of enzymes now decreases. This symbol is nothing but decreases when the temperature or pH actually decreases or increases than the optimum range. Okay, so activity of the enzyme decreases when the temperature or pH drops to below the optimum temperature, or it could be above, that is nothing but beyond. Okay, above the optimum temperature, that's it, or pH. Got it? So enzyme work at optimum temperature. If the temperature or the pH value is decreased or increased, obviously enzyme cannot work. So what's going to happen to the enzyme? The enzyme automatically shuts down. This is it, very simple. Okay, for example, you can just write this also. The low temperature makes the enzyme inactive. So the low temperature, that is lower than the optimum temperature, makes the enzyme inactive. It cannot bind to the substrate. Got it? The active site is now closed for the substrate. Or one more thing, the high temperature, the opposite one, makes, or we can easily write this, or destroys enzyme. Very simple. Egg is also a type of protein, right? When you boil the egg, what is going to happen to the egg? It automatically becomes from liquid to the solid state. That's it. That is nothing but the proteins which is present, albumin and globulin present in the egg, actually undergoes denaturation. The same thing here. The enzyme made up of protein, actually, when temperature is increased, which brings denaturation of the polypeptide chain in the enzyme, and as a result, for example, this enzyme, and when the temperature is high, automatically see it undergoes structural changes like this. Now, can a substrate bind to the active site? No, it's not possible. This is nothing but the enzyme's destruction. When you increase the temperature, when in high fever, when whatever the enzymes present in the body, they get destroyed. That's what, like, if the temperature goes beyond 40 degrees Celsius, that is called as high fever, automatically enzymes present in the body get destroyed. That could be very fatal for the organism or human being. Got it? The human being may die. This is it. Done. I'll give you a graph here if you want. Okay, so this is actually a simple graph, and this is nothing but temperature. Got it? And this one, let me write enzyme activity. The y-axis is, this is y-axis, and this is x-axis. This is the temperature. So see, 0 to, let us say, like 40 degrees, simple. Okay, and in the graph we can write this like this. This is the starting point. See what's happening to the enzyme activity. See, it's increasing now, right? Increasing, now starts dropping. See why? See, this is optimum temperature. So in the optimum temperature, enzyme activity is very, very high. Optimum temperature or optimum pH. Okay, so this is very simple. So this is now optimum temperature. So it is understood that in the optimum temperature, enzyme activity is very high. Okay, it drops down when the temperature is increased. And here also you could see that enzyme activity is very less when the temperature is low or below the optimum temperature. Simple. Okay, or we'll just draw one more graph here for the pH. So for the pH, and here what I should write? pH, and here what should I write? Enzyme activity. Correct? Similar. Now this is optimum pH. See what's happening now. The enzyme activity is very high in the optimum pH. When the pH value is increased, what's happening to the reaction? It is going down. The enzyme is now shutting down. So this is now, this point is actually called as optimum pH. So this pH value is optimum for the enzyme to work. Okay, so optimum pH. So this is actually the thing to understand. So that is the first factor affecting the enzyme activity. Now let me just talk about the concentration of the substrate. Okay, so we are done with these two now. Inhibitors, I'm going to talk about it later after this concentration of substrate concept. Okay, the second important factor which affects the enzyme activity is concentration of substrate. Concentration of substrate is actually our second concept, which is the factor which affects enzyme activity. Okay, see, let me just write this point actually. When substrate concentration, when the substrate concentration increases, see, okay, let me just write this. This is now our enzymes. So we have got how many enzymes here? We have got around 4, 5. Okay, let us just put as 5. Let us assume that we have got 500 enzymes here. Okay, and let me just put the substrate here. See, according to the statement, when substrate concentration increases, okay, so 1 2 3 4 5 6 7 8 9 10. So we have 10 substrate. So let me just write this as 1000 substrates. Simple. 500 enzymes, 1000 substrates are there. Got it? So here the substrate number is more than the number of enzymes, correct? Okay. In the initially, when you start adding the substrate like 400, 500, 600 substrate, automatically all these enzymes now start binding to the substrate, correct? Now 500 substrates are bound to 500 enzymes, but still we have 500 substrate which are left actually, okay, because they don't have any enzymes. All the enzymes are now busy. Okay, so that's what actually the statement is saying. When substrate concentration increases, initially the velocity of enzymatic reaction also rises at first, correct? So the chemical reaction automatically speeds up when all these enzymes starts to bind to the substrate, because we have free enzymes here, 500 enzymes, and we have 1000 substrate. All these 500 enzymes starts binding to these 500 substrates. Automatically the velocity of the reaction, what is velocity? The speed of the reaction increases at the first phase initially. Got it? Okay. Now after this, what if I start adding more substrate? We don't see any change in the velocity of the reaction. Why? Because all the enzymes are busy now. They are taking care of the chemical reaction. Got it? So it is not going to affect the chemical reaction now, because we have all the enzymes which are busy now, which are already bound to the substrate. So this is it. So this is nothing but concentration of the substrate saturates, or enzyme saturates. Simple, enzyme actually saturates. Okay, so we just write this point actually, that is during, because I want you to write this, during this reaction reaches maximum velocity. Okay, so when the reaction reaches the maximum velocity, we just call this as Vmax. Please note this word. What is Vmax? Velocity at maximum level. That's it. So when all 500 enzymes bound to 500 substrate, the velocity of the reaction is very, very fast. Maximum velocity. That is called as Vmax. Got it? Okay. That means all enzymes are busy. In a simple word, we can say that all enzymes are busy. That's it. Okay. The enzymes are not free now. But now, let me just write this also. But we cannot go beyond Vmax. Can you tell me why we cannot go beyond the level of Vmax? Why? Because there are no free enzymes actually. That's it. Okay. So we don't have any enzymes now. So we just write the answer. No enzymes are free to bind to remaining substrate, correct? So we don't have any enzymes to bind to the remaining substrate. What are the remaining substrate we have now? 500 extra actually. So they are actually now waiting for the enzyme to bind. Got it? So we cannot go beyond Vmax. That's it. So this is very simple now. So if you increase the concentration of the substrate, it is not going to increase the Vmax. Yes, why? Because all enzymes are now saturated. They are busy now. So this is what actually the thing you need to understand. Okay, so even if you add as many substrate, but Vmax value cannot be exceeded. If you can just write the point actually, because enzymes are now saturated. Done. I'm going to give you a graph of this actually, that is a substrate concentration graph. Please note this. Okay, so we just draw here. Please note this graph. This graph is very, very important actually to understand enzymatic inhibitors. That is actually our fourth factor which affects enzyme activity, that is inhibitors. Okay, so this graph is very important. Let me just draw the graph and I'll tell you what is this. This is our y-axis and this is our x-axis. Okay, so this is the start of the reaction. Here no need to understand that potential energy, that is actually not required for this actually concept. And this is the Vmax, that is velocity of the enzymatic reaction, that is actually the maximum value or the maximum velocity. Okay, so this is the Vmax. Okay, so now initially I have got a lot of enzymes which are free now, which starts binding to the substrate. Okay, slowly now the reaction rate or the velocity of the reaction is increasing. It's going, going, and now all the enzymes are now saturated, correct? They are busy now, they are bound to all the substrate. Now from this point onwards it cannot exceed, actually this is the Vmax, the maximum velocity of the reaction, because all enzymes are now busy. So this is what actually you need to understand. See this graph. So Vmax value cannot be exceeded even though if I start adding substrate, because all enzymes are busy. Simple. Okay. And now this is actually the state of the reaction. See, this is the initial state of the reaction when all the enzymes are starting to bind to the substrate, correct? Okay. So here, this point is actually velocity of the enzymatic reaction, that is Vmax by 2. What does it mean? This is half Vmax. So I just write this here. Vmax by 2 is nothing but half Vmax. Got it? So this is actually the half value of velocity, the maximum velocity, halfway. Got it? Okay. So here let me just draw one small graph like this. Okay. So this is actually half velocity. Okay, half maximum velocity during a chemical reaction where 50% of the enzymes are busy with the substrate. Remaining 50% of the enzymes are now trying to bind to the remaining substrate. Got it? So this is actually the half maximum velocity of the reaction. Got it? Okay. And this can be this one on the x-axis. I simply can write this as Km. Okay, Km is the one value. Okay, when the enzymes are busy, half enzymes are busy during the half maximum velocity. Got it? Okay, when the reaction is undergoing halfway, that is actually Km value. This is the Km value. Okay, so this is, you need to remember. I should have mentioned this. This is actually nothing but substrate. X-axis is nothing but substrate, and this one, the y-axis, is nothing but the velocity of the reaction. So velocity of the reaction, and this is the substrate, and ultimately you get a graph like this. Okay, so this is what, remember, the chemical reaction cannot go beyond Vmax value. Okay, but here this is the halfway of the chemical reaction, and you assume that it's going to go beyond Vmax, but it's not going to go. Got it? But we need to take this value. This is the Km value. Let me just write the chemical reaction, how exactly it takes place. When enzyme binds to the substrate, it forms this. What is it? Enzyme substrate complex. So this is actually the first step. So what's happening here actually? Okay. Second is actually you are going to get from ES, you get products and also enzymes. Enzymes are getting free now. Got it? So product and enzymes. Done. Now let me write, what is this Km? So Km is nothing but concentration of active mass of the substrate. Concentration of the active mass of the substrate, correct? Okay, substrate at which rate of reaction is half Vmax. See, when the reaction is halfway, you get a value actually. Okay, so very simple actually. In this point you see around 50% of enzymes bound to 50% of the substrates, correct? So you get a value here, that is Km. Km is nothing but active mass of the substrate. Okay, how many substrates are now bound to how many enzymes when the reaction is in its halfway? Okay, so that is half Vmax. Okay, so next one, let me write this. During this reaction, when enzyme binds to the substrate, okay, this reaction can be reversible. That's what I have put the opposite arrow mark here in the two direction. You get enzyme substrate complex, and ultimately you get product plus enzymes. Enzymes are now becoming free actually, so that they can bind to the substrate. Okay, so here I just want to write the values Kf and Kr. I'll tell you what is this Kf and Kr, and here we just write Kcat. Okay, so let me write that what is that Kf is. Kf is nothing but rate constant for formation of ES. What is the speed of that particular reaction between enzyme and substrate. Got it? So rate constant for formation of ES. So that is Kf. Okay. Next one is Kr. What is this Kr? Again, rate constant for dissociation. Now enzyme substrate complex should dissociate, correct? Because the products are formed from substrate, but they are now separating. For dissociation of enzyme substrate complex. So this is Kr. So Kf is important rate constant. What is the speed of the binding between enzyme and substrate is taking place? That is nothing but Kf. Kr is nothing but it is the rate constant. What is the speed of dissociation of enzyme and substrate? Okay, both are different value. Formation of enzyme substrate complex may take 0.1 second, but dissociation of substrate from enzyme takes 0.2 second. So that is actually the different rates. Okay, it varies, and both varies because what type of bonds are present in the substrate. Now don't worry about it, it's not required. Actually in higher classes you're going to study about this. Fine. Then we have left with this Kcat, when enzyme substrate complex actually producing the product. Okay, so that is nothing but Kcat is nothing but rate constant for the chemical. That's it. Okay, so this is nothing but Kcat. So here the bonds, how many bonds are present in the substrate to produce the products? Okay, what is the effort required by the enzyme to break bonds in the substrate is nothing but Kcat. So rate constant for the chemical. More the bonds, if the chemical is more complex, obviously the effort taken by the enzyme is more. That's it. If the substrate bonds are very simple, okay, if the substrate is very simple chemical, then obviously enzyme takes very less time or less effort to break the bonds in the substrate to produce the products. That is nothing but Kcat. It varies from chemical to chemical. Got it? So this is actually the simplest way of understanding. Hope you understood this. Okay, there is no confusion here. Please understand what is Vmax. Vmax is nothing but it is the maximum velocity of the chemical reaction. Got it? Okay. How? When all the enzymes present in that particular tissue are used up or busy, when they bind to all the substrate, that is actually maximum velocity. Even if I start adding more substrate, that is when I increase the concentration of substrate, it is not going to affect the Vmax. Vmax value cannot be changed. Vmax cannot be changed. Please note this point. Why? Because all enzymes are saturated. They are busy now. Okay. This one value should be noted. That is actually half Vmax. What is half Vmax? It is actually halfway of the chemical reaction. Okay. When some enzymes, 50% of the enzymes are busy now. They are not saturated. Got it? Okay. You may think that the value could go beyond this Vmax. No. Actually, at some point, all enzymes should be busy when they bind to all the substrates. That's it. Okay. So this is actually simplest way. If you want, you can just recall it. Not necessary actually. If you write it and recall it just before the examination is fine. Okay. Otherwise, you cannot remember this for longer time. Done. Okay. So now we will talk about the enzyme inhibitors. Now our last factor which affects the enzyme activity, that is enzyme inhibitors. Enzyme inhibitors. Please understand the meaning of inhibitors or inhibition. Very simple. It's nothing but a type of chemical or substance which is going to interfere the chemical reaction and slows down the chemical reaction, or sometimes it could stop the chemical reaction. Very simple actually. When you are running here, and obviously there are so many factors, right? You could be like running uphill. Maybe you are running the mountain, in the mountain uphill direction, or sometimes actually it could be downhill. Correct? In the downhill, obviously your speed will be more. In the uphill running, obviously your speed will be very less. This is one factor. Or sometimes actually there are so many things. Actually there could be thorns, there could be spines, or there could be rocks, boulders, whatever. Okay, it's going to interfere your process, that is running. Okay, obviously like it could bring the speed low. That's it, or down. Got it? So this is actually inhibitors. Here, let me just tell you what is this chemical or enzyme inhibitors. Okay, so let me just write what is it. when any chemical, any chemical, okay, any chemical binds to the enzyme. Where it binds to the enzyme, it could be active site, or maybe there could be some other sites also. Let us see that. Okay, when any chemical binds to the enzyme instead of substrate. So when enzyme, actually, they should bind to particular specific substrate, but instead of that substrate it binds to another chemical. Obviously it's going to affect the enzyme activity, so it shuts off enzyme activity. Got it? So this is very important actually. When any chemical binds to the enzyme instead of substrate, it shuts off enzyme activity. So this is called as enzyme inhibition, or in a simple word I write inhibition. This is it. Automatically enzyme reaction is now stopped. Okay, so here let me just tell you, this is now the enzyme and this is actually the actual substrate, so that it's going to produce enzyme substrate complex, ES, to produce the product. So I always write like this because enzyme should be there. Okay, ultimately enzyme dissociates from the substrate, and ultimately substrate produces products after transition state. Okay, after breaking the bonds in the substrate, it is going to produce new products. This is actually the actual products to be produced. Got it? But now what if we have another chemical very close to the substrate? They are acting like substrate analogs. So it is another chemical, not this chemical. Actually the substrate is different, so it is a substrate analog. What is the meaning of analog? Analog is nothing but duplicate. Okay, so duplicate, so which actually mimics or resembles the actual substrate. This is actually the actual substrate which has got a chemical structure, whereas this is actually not actual substrate, but which resembles this actual substrate in its appearance. It's very easy for this chemical, that is inhibitor, to fool this enzyme, so that enzyme thinks that this is the actual substrate and binds. Instead of binding to the actual substrate, it binds to a fake substrate. Got it? Now you can imagine what kind of product it is going to produce. So it's going to affect the enzymatic reaction. That's it. Very simple. Okay, so let me just write the types of inhibitions. So this is very important. There are three types of enzyme inhibitors. So this is very important. You understood what is inhibition? Inhibition is nothing but when a substrate binds to the enzyme and slows down the chemical reaction. Okay, so this is nothing but inhibition. Okay, types of chemical or enzyme inhibitors. The first one, very important, competitive inhibition. So what is this competitive inhibition? Very simple here. This chemical, so the chemical which is not actual substrate, the chemical is substrate analog. What is the meaning of analog? The duplicates, which looks like actual substrate but they are not actual substrate. Okay, so this chemical analogs, that is is like fake substrate. Now this chemical now binds to the active site. So that means it is easy to understand that competitive means competition. Competition is occurring between whom? The competition is occurring, let me write here, the competition is occurring between substrate and substrate analog. Duplicates, please understand. Substrate and substrate analog. For what? For active site. That's it. Okay, so for this one active site, two different chemicals are fighting. Okay, actually this substrate should bind to the active site of the enzyme to take up or to initiate the chemical reaction, but substrate analogs going to compete with the substrate and binds to the active site. Okay, it's nothing but fooling the enzyme. That's it. So that enzyme actually thinks that the substrate analogs are real substrates, and it's initiate the chemical reaction. That is actually it starts going of taking care of the chemical reaction, but the product will be different, or the quantity of the product will be very less. It's going to affect the enzyme activity. Okay, so here the possibilities. So let me just give the diagrammatic representation. This is the actual enzyme and this is the active site, and let us use the same color. This red color is now the substrate, the real substrate, and we have substrate analog here. This is substrate analog, the black color one. Okay, actually this enzyme should bind to this red color substrate, but as this black color substrate is mimicking this actual substrate in its structure, chemical nature, it's going to bind to the active site. So you are going to get this picture. The substrate is now different, correct? So this is competitive inhibition. So this particular substrate analog is competing with this actual substrate for the active site. That's it. So this is competitive inhibition. What's going to happen to the reaction now? It slows down, or it could be shut down also. Okay, you are not going to get the actual products also. This is it. So possibilities here, in this case, the possibilities, please note this point, very important. The possibilities, the first possibility is enzyme and substrate complex can be formed. Yes, right? ES is can be formed. Yes, when the substrate actually wins race with this substrate analog, it can bind to the active site of the enzyme and real reaction runs. So ES is also possible here. At the same time, when this substrate analog wins the race with this real substrate, so it's going to produce EI. That is also possible, right? What is EI here? When this one is inhibitor, right? Chemical, this is the actual substrate. So ES is possible. Even EI is also possible. If a substrate is strong, like inhibitor is stronger than a substrate, obviously it's going to bind to the enzyme. So EI is also possible. Simple. Okay, so note this point. So EI is also possible. But is this possible? ESI is possible. How? Okay, let me just tell you how. So enzyme here and this one EI, correct? Possible. Okay, enzyme here, and yeah, I should use red color now. This is real substrate. That is also possible. EI is also possible. ES is also possible. But what about the third one? Sorry. Yeah. This is possible? No, it's not possible, because this inhibitor is competing for the active site. It cannot bind to the other site of the enzyme. So ESI is not possible. So note this point. Okay, in competitive inhibition, ES is possible, EI is also possible, but ESI, when substrate as well as inhibitor binds to enzyme, is not at all possible. ESI is not possible. Got it? Because they are competing for the same site, that is active site. Okay, so inhibitor cannot bind to any other site of the enzyme. Done. Okay, I'll give you a good example here. Let me just turn this page. So the good example for this is very important. Okay, example number one. Let us just write example number one. I just write succinic acid. It is nothing but substrate now. The normal, the actual substrate. Okay, so succinic acid now produces fumaric acid. This is the actual product, fumaric acid. When you are going to add succinate dehydrogenase enzyme. Okay, so succinic acid, when succinate dehydrogenase binds, so this is the substrate, this will bind to succinate dehydrogenase active site, and ultimately a product is formed, that is fumaric acid. This is the normal reaction. It's happening in the body. Okay, now we have competitive inhibitor which looks like succinic acid, but they are not succinic acid. Okay, that is malonic acid. The malonic acid structurally and chemically looks like succinic acid, but are they succinic acid? No. So it's inhibitor. So just write inhibitor here. Okay, so now enzyme is same, succinate dehydrogenase, which now fooled by the malonic acid. Now malonic acid binds to the active site of the succinate dehydrogenase and you are going to get the same product. The product is not changed. You are going to get the same product, but here we are getting like hundreds of fumaric acid. Here we are getting 2 fumaric acid. That means this is inhibition. Yes, this is inhibition. The enzyme activity is affected by this inhibitor. So this is competitive inhibitor. That means actually malonic acid is competing with succinic acid for the active site of succinate dehydrogenase. Done. So this is one simple example here. Okay, so I'll give you the graph of it. So for this one, let us name it as A and this reaction as B. Okay, for A reaction, that is actual reaction, let me write this y-axis, velocity of the enzymatic activity or reaction, and this is the substrate. What is the substrate? Succinic acid is the substrate, correct? Okay, so what is this? Vmax. Now you are getting my point. Okay, so this is half Vmax. I write Vmax by 2. Okay, so now during this, succinate starts binding to the enzyme or enzyme binds to the succinate. Okay, so now the reaction slowly catches the speed and ultimately reaches the Vmax. From this here onwards, it cannot go beyond Vmax. This is what we understood earlier. Okay, and now this is halfway of the reaction, and see this, what is this now? Km, correct? So this is Km now. Okay, now I'm going to draw this second graph for this malonic acid. See, you could see a bit change in the graph. Okay, so this is malonic acid and this is velocity of the reaction, and this is, we know that V max by 2. Okay, and this one is V max, correct? Okay, let us just see that now when malonic acid competes for the active site of the succinate dehydrogenase enzyme, and you could see, so there is a slight change in the graph. See, the Vmax value remains the same in both the reaction. Vmax will be same because they are chemical analogs. Okay, so enzyme is getting fooled by the malonic acid, so that Vmax never changes. It is unchanged. Vmax is unchanged, but you could see the value of Km now. So in the halfway of the reaction, you could see that the Km value is slightly different. Can you see? See. So what has happened to the Km value in the second reaction, malonic acid? Km value is increased. So please remember, in competitive CI, in competitive inhibition, there are two important things to note. The Vmax value remains unchanged, correct? In both the cases, Vmax value is same, but the value, that is Km value, is increased or increases, correct? In the malonic acid case, you could see that the Km value is very high actually. Got it? So these are the 2 points you need to mug up for competitive. It is going to be changed actually for the second type of inhibition. Okay, so remember this for competitive inhibitors. What is competitive inhibitor? Competitive inhibitor is nothing but when another chemical which acts or mimics the actual substrate in its structure as well as chemical nature and tries to bind to the active site and changes the velocity or Km value of the chemical reaction. That's it. Okay, so this is what competitive inhibitors. Now for your competitive part, you could get this question. Very important actually. Okay, so name the competitive inhibitor of succinic acid. Which is the competitive inhibitor of succinic acid? Malonic acid. Remember that. Note this point. Very important. I'm telling now itself. Okay, so note this point. You can write it down. The next example, I just want to give you the second example. That is another chemical, that is aramino benzoic acid. Okay, there is one acid actually. For this we need one enzyme. This is the normal substrate actually for the enzyme. Folic acid synthetase. Folic acid synthetase is actually the enzyme where the substrate for that is aramino benzoic acid. Okay, to produce the product called as folic acid. So this is our actual reaction, normal reaction. Okay. But there is a competitive inhibitor for para-aminobenzoic acid. What is that competitive inhibitor? That is sulfur drugs. Please note this point. You may get a question. Which is the competitive inhibitor of para-aminobenzoic acid? The sulfur drug. Sulfur drug looks similar to para-aminobenzoic acid and it's going to fool the folic acid synthetase. So the enzyme is same. Correct? So synthetase. And now sulfur drug binds to the folic acid synthetase active site and it's going to produce the same folic acid, but the number of folic acid in the second case produced will be very less. That is, reaction, chemical reaction is affected. Simple. Okay. Here again I'm going to tell you ES is possible, that is folic acid and parabenzoic acid complex is possible. Yes, ES is possible for this also, right? Succinate dehydrogenase and succinic acid, ES is possible. Yes, possible. EI is possible. Yes, when succinate dehydrogenase bind to malonic acid, that is inhibitor, that is enzyme and inhibitor is also possible. Yes. ESI is possible? Never. You cannot see succinate dehydrogenase binding to succinic acid and malonic acid at the same time. It's not possible. So ESI is not possible. Done. Remember, Vmax is unchanged for this, whereas Km value increases. Km value increases for what? When inhibitor binds to the enzyme. Done. So this is, hope you understood. This is very, very important concept. Okay, that's what I took my time. Done. Now we will talk about another type of inhibition, that is non-competitive inhibitor. Okay, so let me just change to the next page now. The second type of inhibition. Second type of inhibition is non-competitive inhibition. Non-competitive. Here competition is not taking place. That's it. Non-competitive inhibitor. Okay. First one is competitive, second type is non-competitive inhibitor. Here, the inhibitor. Now in this case, the inhibitor is not the chemical analogs. They are not the duplicates. They are different. Okay. But still they can bind to the enzyme, but not to the active site. Other than active site, they bind. Okay. So inhibitor now actually attaches to enzyme, but not to the active site, that is other than active site. Actually here the real chemical, the real substrate will bind to the enzyme's active site, but inhibitor will bind to some other site other than active site in the same enzyme. Okay, for example, let me just draw the enzyme, and this one is the active site of the enzyme, and okay, so let me just use the blue color itself, and this is actually the actual substrate, is the actual enzyme. Okay, and we have the inhibitor here. We have inhibitor here. In the competitive inhibition, we know that here the chemical is competing with the substrate for the active site. But in this case, non-competitive. Here the competition is not taking place between substrate and the inhibitor. So I'm just using a plus symbol. That means the substrate inhibitor, that is inhibitor is not similar to substrate in its chemical nature as well as structure. It's entirely different chemical. Okay, so it's going to bind to some other site. So it's going to bind to some other site of the enzyme. See here, it's bound to some other site other than active site. Got it? So here you can easily say that ES is possible. Yes, when substrate bind to active site of the enzyme, ES possibility takes place. EI takes place. Yes, even and this inhibitor can bind to the enzyme other than active site. So EI is also possible. Okay. Is this possible? ESI, enzyme, substrate plus inhibitor. Three things. Enzyme, substrate and inhibitor. Possible? Yes. In this case it is possible, but in competitive inhibitor it is not possible. Why? Because the site both are competing is active site, that is same. But in this case the inhibitor is not competing with the substrate for the active site. It can bind to any site of the enzyme other than active site. So three things are possible in this case. Yes? Okay. Now I'll give you the graph. So here you know that this is y-axis, that is velocity of the reaction, and this is substrate, x-axis. Correct? So velocity and this is substrate. Okay. Now For simple, when a normal reaction is taking place, so this is nothing but maximum velocity of the enzymatic reaction, that is Vmax, correct? We know that, and we know that this is half Vmax by 2, okay, halfway of the chemical reaction. Now when a substrate without inhibitor, so let us assume that inhibitor is absent, only substrate is there, substrate is going to bind to the enzyme, and automatically what's happening here, the Vmax finally achieved. This is normal, got it? So normal, the enzyme activity is normal actually when enzyme and substrate only are present, when both are present. Actually here inhibitor is absent, okay? So we are going to get the Vmax value. So what is that Vmax value? So this is the Vmax, that is nothing but Km, correct? Okay, see here Vmax remains same, Km also remains same here, okay, for first case. Now let me just sorry, that's not that. In the second case, let us assume that when inhibitor binds to ES, when already substrate is bound to enzyme, now another chemical, that is inhibitor, binds to that, okay? So what's going to happen now? See, this is Vmax. So Vmax value now decreases when inhibitor binds to the enzyme, okay? But see here, the Km value remains the same, Km remains same. So I can write for non-competitive inhibitor, Vmax value increases or decreases? See, decreases when inhibitor binds to the enzyme, okay? So Vmax value decreases, but Km value remains same, or in the bracket you can say that Km value remains unchanged, got it? So this is what you need to understand, please understand this, got it? Okay, in the first case, competitive inhibition, Vmax value remains unchanged, but Km value increases when inhibitor binds to the enzyme. In the second case, that is non-competitive inhibition or inhibitor, here the Vmax value decreases when inhibitor binds to the enzyme, Km value remains same, that is unchanged, got it, when inhibitor binds to the enzyme. This is what you need to remember. Example is, let me just write, I think you have recently studied about electron transport system. In that, cytochrome oxidase is there, enzyme is there, okay? So we know about cyanide poisoning. When any person takes cyanide, it's a chemical, it's an inhibitor actually. When cyanide is taken, ingested, that is taken orally, so it's going to bind to the mitochondrial enzyme called as, that is enzyme present in mitochondria, that is cytochrome oxidase. When this cyanide actually taken orally, it is going to bind to cytochrome oxidase enzyme, okay? Already cytochrome oxidase already bound to its actual substrate, that is enzyme substrate is already formed. Now the inhibitor, that is our cyanide is going to bind, so ESI is possible now, and ultimately it entirely stops the reaction, chemical reaction is stopped, and ultimately cytochrome oxidase cannot work, and ultimately mitochondria cannot produce ATP, and ultimately you see death. So it kills the organism. So cyanide poisoning. So this is very simple example for non-competitive. Here cyanide is not competing for the active site of the cytochrome oxidase enzyme. Got it? It's going to bind to some other site in the cytochrome oxidase and just inactivates this cytochrome oxidase enzyme, and as a result ATP cannot be synthesized during electron transport system or chain. Done. It causes the death of the organism. So this is what actually the second important type of inhibition. I'll go to the third type, the last type actually, that is uncompetitive. It's not required, but please write on this also. Uncompetitive inhibition. Okay, uncompetitive inhibition. Please write on this. Inhibitor actually. When inhibitor binds to the enzyme, in this case also same, other than active site, when enzyme already bound to substrate. Okay, so when, almost similar to non-competitive, but there is a slight difference. When enzyme already bound to substrate. Okay, so in this case, the thing is, let me just tell you, so this is now enzyme, okay, and I'm using black color. This is the actual substrate. So now you see enzyme substrate complex. Okay, when enzyme substrate complex is formed, now the chemical, that is inhibitor, comes and binds to some other site in the enzyme other than active site. In this case, please remember, like first inhibitor can bind, then substrate can bind, or substrate first binds or inhibitor later binds. Okay, so three things are possible. But in this case, for the inhibitor to bind to the enzyme, we need ES. So that's what, when inhibitor binds to enzyme other than active site, when enzyme already bound to substrate. This is very, very important. Already enzyme bound to substrate, then only the inhibitor can come and bind to the enzyme. Okay, so now ES becomes ESI. Yes, you are right. Correct. So here ES is possible. Yes, correct. ESI is possible. Yes. Remember that enzyme substrate both are required for the inhibitor to bind. Okay, inhibitor cannot bind to enzyme if the enzyme is not bound to substrate. So we need this important thing. Okay, enzyme substrate complex is must for the inhibitor to bind to the ES. Is this possible? No, it is not possible. In the non-competitive inhibitor, EI is possible, correct? So substrate is not required. But in this case, EI formation cannot take place. Why? Because inhibitor cannot bind to the enzyme's active site if enzyme is not bound to substrate. So this is not possible. So these are the things you need to remember. You need to like practice. Got it? Okay, no confusion here. I will not draw the graph actually. I'll give you the value. Please write on this. Vmax value plus Km value both actually decreases in this case. Okay, so please write down this. Vmax value and Km value both decreases when ESI, this is must actually. Got it? For the inhibitor to bind to enzyme, substrate should be already bound to the enzyme. This is compulsory. So ESI is the state. So in the ESI, Vmax value, Km value both decreases. Okay, one simple example. I think I don't want to take a new page. Okay, so I'll write here the example for this. Simple. Hydrogen ion. Hydrogen ion is the inhibitor. Okay, it's an inhibitor of alpha amylase enzyme. So note this point. It could be asked, not important actually. Okay, hydrogen ion is the inhibitor of alpha amylase enzyme. Got it? Alpha amylase should bound to its actual substrate. Then only the hydrogen ion can bind to alpha amylase. Okay. Otherwise, hydrogen cannot bind to alpha amylase. When alpha amylase is free, there is no substrate. Okay, so hope you got this point. That's what I have written here. ES is possible, ESI is possible, but EI cannot be seen. EI is not possible. This is it. Okay, Vmax value, Km value both decreases in this case. Hope you got this point. Done. Okay, now we'll discuss about the classification. I think I'll take one more video session.